Hi there, I'm Fritz, a research scientist from OZ Technologies, and today I want to talk to you about data transformations for effective visualization of single-cell embeddings. But before I start, just a quick disclaimer that although several authors and myself are affiliated with OZ, the work was conducted at Fritz Lekschas, is published in a peer-reviewed journal, and can be accessed freely and openly on the web. So, in a nutshell, with the data transformation that we've developed, one can create embeddings that provide a higher resolution view of computationally derived clusters that we refer to as cell phenotypes that otherwise often blend together visually. So for instance, here on the right side, we can see how the distinct colors which represent these cell phenotypes also cluster visually while being hardly indistinguishable on the left. But what are cell embeddings? So a single cell embedding is a lower dimensional representation of high dimensional single cell data. Say we have a table where each row represents a cell and the columns are different cellular features of the proteome, genome or epigenome of the cells. An embedding is a representation of this data in 2D or 3D that tries to represent the high dimensional topology as best as possible. The reason people visualize such embeddings is often to obtain a complete overview of all cells and how they relate to each other, to generate hypotheses, annotate clusters, or compare cell types. Now, there are various linear and nonlinear methods to create such an embedding. And here are three real-world examples, showing, for instance, cell surface antibody expression, gene expression, and chromatin accessibility peaks-based embeddings. Now, while such embeddings provide an exciting opportunity for exploration, there's still some challenges with the resulting visualizations that relate to specific or rare cell population, as well as batch effects of multiple samples. So for instance, are we interested in just broad patterns that reveal general cell types? Or do we want to be able to visually differentiate more specific cell types, as indicated here by those colors? Also, when embedding multiple samples, how shall we handle batch effects? Are we actually interested in the difference between the two samples shown here? Or do we rather want to focus on the cellular differences? And so in general, an overarching question is, are we using the embedding to explore or to explain clustering results? And the reason why it can be challenging to achieve both is because there can be a disconnect between the data used for embedding and the data used for clustering. To tackle these challenges and make the visualization more a representation of the clustering, we have developed a method to annotate the raw expression levels. Without going into any technical details of our FAUST method here, the high-level idea is that for each cellular feature, like these surface markers, our method annotates the modes with a human interpretable level, like positive or negative, for these bimodal expressions that we see here on the right, for instance. And doing this allows us to easily group cells into meaningful and interpretable clusters. So if you're curious about the details, please see the paper that I've linked at the end of my talk. But what's important for the visualization is that each cell here is fully annotated. And so if we give each fully annotated cell a unique color, we can see here that the color information hints at a lot more details than what is being revealed by the structural information of this embedding plot. But how can we actually detect these clusters, not just with colors, but also structurally? And so this brings us to our annotation transformation. The transformation approach consists of three simple steps. First, remove extreme values, then normalize the expression ranges, and finally align the expression ranges. To give an example, say we look at T helper cells that are CD3 positive, CD4 positive, and CD8 negative. First, we windsorize the marker expression to effectively remove outliers. Next, we normalize the expression value of each marker to have zero mean and unit variance. And finally, we translate the expression ranges based on the expression level to align the markers. And so if we transform the data in such a manner before embedding it, we are able to reveal clustering information structurally and not just through color, as you can see here on the right, using, for instance, a standard UMAP embedding. We can achieve the same effect not just with UMAP. Here we see virtually the same result when embedding the data with t-SNE. And we can even reveal the same information when we use a variational autoencoder model, which on its own doesn't show much, But the point is, the structure is revealed by transforming the data first, not by picking or tuning any specific embedding method. Beyond revealing structure, our transformation also ensures that cells of the same cluster are placed more coherently. Here I'm highlighting two cell phenotypes that only differ in their CD8 expression and strongly overlap in the untransformed embedding, while being well separated in our transformed embedding. And so when we look at the CD8 expression, we could identify a trend in the untransformed embedding, but it's hard to perceive it as a unique cluster. Interestingly though, CD8 expression has been shown to define pre-activated, tumor-infiltrated CD8 positive T cells. So it could be biologically relevant to perceive these two cell phenotypes as distinct clusters. For joint embeddings of multiple samples, in this case a tumor and a healthy tissue sample, our transformation entirely removes any batch effects, which makes it easier to really identify coherent cell phenotypes detected with our clustering method. In an untransformed embedding, the cells that I've selected here actually belong to the same cluster. It appears spread out and disjoint. And such coherence can be important or useful for inspecting rare cell populations. Now I want to conclude with 3 takeaways. First, if there's one thing you remember from this talk, then I hope it is that when you create an embedding, do the data work and optimize your data before tuning the embedding method. Second, use a data transformation that is closest to your objective. So for instance, our annotation transformation helps to convey the clustering results much better than the untransformed data. But that doesn't mean that you should use it for all tasks. And finally, even though the annotation transformation works well with our clustering approach, it's not strictly bound to it. You can use any method you like to derive the expression level annotations.